Okay, so thank you, Mayalda. Third speaker of this session is Wayne Hubbell from UCLA. He will tell us about his work on Rhodopsin structure and dynamics and function with the Gobind group. And the title of his talk is Gobind Khorana and the Central Dogma of Receptor Activation.
So am I on the air? Okay. Well, the process by which biological organisms convert physical and chemical stimuli coming from their environment and convert them into a cellular response has intrigued chemists and biologists and even physical chemists for a long time. This process is generally known as signal transduction. And the first signal transduction system to be understood in any molecular detail at all was that of the visual system, the photoreception system in the vertebrate retina. And in this process, there's a molecule called Rhodopsin. It absorbs a photon of light, and it triggers a cascade of events that ultimately results in the generation of an electrical signal that's sent to your brain to give you an image. Now, in the late 1980s, biochemistry by which this happens was worked out. And let's look at that quickly here on this slide. So here is a disc membrane. This is an intracellular organelle in your photoreceptor. Now, within that membrane is a transmembrane protein. This is the rhodopsin photoreceptor I just mentioned. It has 11 cis retinalis, a chromophore. And when light is absorbed, it isomerizes all trans, and that's all light does in this event. But nonetheless, it triggers a transformation in rhodopsin, we'll call the rhodopsin switch, to some new conformation. And that new conformation is now recognized by a molecule called transducin that's floating around in the cytoplasm. It's a heterotrimeric G protein. And transducin recognizes this new form of rhodopsin, binds to it, and then binding energy switches the alpha subunit to a new state which sort of boots out around GDP. And in the presence of GTP, which is always around, it occupies that empty site. That interaction switches the alpha subunit to yet another conformation called the activated conformation, which then diffuses around the cell until it finds the phosphodiesterase enzyme, which it turns on, switches it on. And that degrades cyclic nucleotides in the cell, cyclic GMP to GMP, and that turns out to close ionic channels in the plasma membrane. Now, you know when you close the ionic channels, you block an ionic current. And when you change the current through a membrane capacitance, you get a voltage signal. And that's it. This thing is a light to voltage converter of enormous amplification. Now, about this time, Gobind and I shared a common interest, and that common interest was to understand how this works. What's the means by which this molecule is activated by a photon of light? We knew quite a lot about, a reasonable amount about what the structure of this inactive state was from modeling and other things, but we knew nothing about how this thing was activated. Now, at that time, my laboratory was involved in developing site-directed spin labeling, a technique where we put nitroxide spin labels in a protein and use it to determine conformation and changes in conformation, exactly what we wanted to do here. And now, in 1986, at the same time, Wayne Hubbell published the synthetic gene for rhodopsin and its expression in amounts sufficient to do this kind of experiment with spin labeling. So we met at a meeting and we discussed the possibility of using his gene to produce sites that we could place the spin labels in and explore this activation mechanism. And he was enthusiastic, he was willing to proceed. It was the luckiest moment of my life, actually, when he did. And so I'm going to tell you in a second what we learned and a few things about it, but I'll make a few comments about this period, which is intense, let me say. A few other people have mentioned that word in connection with Gobind, and it was true. It was an intensely exciting experience. We published 27 papers on the subject in a relatively short period of time. I would travel back and forth from UCLA to MIT and go to group meetings, and we'd have discussions, and we'd walk, and we'd talk, and I was exhausted when I came back. And once in a while, Gobind would sense that I might be distracted by something, like sleep or food. And he'd say, Wayne, we have to focus. Wayne, we must focus. And I've heard that several times. And he took me into his office and we sat down and we talked. He said, look, it's really simple. And he had this thing on his wall that many of you have probably seen. And he said, Wayne, this is all we have to do. You work, finish, and publish. It's very simple. It's like start at the beginning, go on to the end, and stop. So this was simple. Gobind explained to me that he knew lots of people that could work but never finish. Newton said would work and finish but couldn't manage to publish. And the worst of all, those that worked didn't finish but published anyway. And he said, Wayne, we have to do all of these things and this is how we're going to solve the problem. So from that I learned a lot. I learned to focus. And so what I'm going to do today is focus very specifically on what we did in Rhodopsin and tell you about what the present status of the work is and what the future might be. Hopefully it's before my time's up. Now, let me tell you very briefly about the spin labeling experiment. It's probably familiar to most of you, but what we do is we pick a site in a protein, this is a Rhodopsin, of course, but it's an illustration. We pick a site in a protein, we mutate it to cysteine. Then we react that cysteine with a highly selective methane thiosulfate nitroxide reagent that makes a disulfide-linked nitroxide side chain in the protein. Now this thing is paramagnetic, this nitroxide, and it has an electron paramagnetic resonance spectrum. And you don't need to know anything at all about where that spectrum comes from, but just to appreciate the fact that it's exquisitely sensitive to the environment of the spin labeling protein. So for example, if one were to walk that little nitroxide through that helical sequence one at a time, scan it through, the spectrum would be periodic because of the periodicity of the environment and the helix. So this allows one to determine sequence-specific secondary structure. And one can then put in two nitroxides by the same experiment, the same mutagenesis, and they interact by a magnetic dipole interaction, and from that interaction strength you can get the distance between residues in this sort of short range here. And you can appreciate that if you can determine secondary structure and distances, you can assemble a structural model of something in 3 dimensions at the level of the backbone fold. And many such things have been done. So at the time we started our experiments together with Gobind, this is the state of the art. But I'll just say as a sideline now, some really recent dramatic developments in technology, and I'll comment at the very end of the talk about what's come from them. So some new things. A technique called DEAR, Double Electron Resonance. It allows one now to measure distances over a very long range, up to 60 angstroms, dramatically extending this experiment, and very high resolution measurements. And for me, actually more exciting, are new techniques called pulse saturation recovery, which some of you may have heard of, and high pressure EPR. And this allows us not just to do structure, which comes from here, but to do flexibility and dynamics of proteins. And as you probably know, the view of proteins these days in solution, they don't look like the images you see for crystallography. They're not rigid, but in solution they're highly flexible, moving around all the time. And these motions are critical and central to the function. So if you can't measure the motion, you won't understand the function. So now this moves us into a range in which we can do that. And I'll say at the end just a few comments about this. So now going back to what we had when we started our experiment. So we could do structure. So with Gobind's gene, we put that spin label at more than 100 single sites, marched it around the entire cytoplasmic surface of Rhodopsin, which is the surface that recognizes transduction. And we put more than 40 pairs of spins in to do this distance measurement to map structure. And in a series of papers, actually 4 or 5, which were in one journal back to back, we predicted the structure determined the structure, secondary structure, and some 3D features of that cytoplasmic domain. This is way before the crystal structures came out of high-resolution structure. So let me just put what we did in context of a high-resolution structure. I'll show you. But there are several. The first was in 2000 or 2001 by Kalshevsky's group, and subsequently there was a series of structures. I'll show you the one that comes from Lee and Schertler at MRC. So this is what Rhodopsin looks like. 7 transmembrane helices, and here's the 11 cis-retinal chromophore. These orange lines here are the membrane interfaces. This is the cytoplasmic surface that's recognized by transducin. Now remind you, this is the inactive state we're talking about, the crystal structure. Now these colored sequences you see here are the sequences that we march this nitroxide through one at a time, all the way through there, those 100 residues I was talking about. Now the conclusions from our structure of determination from the spin labeling which preceded this by a lot. And in fact, were an extraordinarily good agreement to the extent that they could be compared, and that was rather extensive. So this is a cross-validation. I mean, it validated the crystallography and it validated the spin labeling to determine the structure. So, but one thing it couldn't do from the crystallography that we could do, and what our major interest had always been, is looking at the evolution of this structure. When you excite it, what happens when you turn on the Rhodopsin switch? And what I'll just tell you is that all of the work that we did on the structuring, comparing the crystallography, so Gobind and I wrote a long review in 2003 that contains all of this information. But we learned a lot of things, but probably the most exciting and had the biggest impact was we discovered what really is the Rhodopsin switch. And that turned out to be an outward movement of this helix by we didn't know what that came out for, but an outward movement of this helix TM6. And Dave Theron's and King Yang were involved in this, Gobind's group, Christian Altenbach and my group, and it was a very exciting discovery. But it was a low resolution measurement at the time. We didn't know exactly how far, we didn't know the trajectory of the helix, and we didn't know whether there were minor movements in other helices. But this was it. This was a benchmark determined the first time that a structural correlative activation had been described. Now let me show you a high resolution measurement now of that motion. So this is using that new technique I just mentioned of DEER. So this is a top view of the Rhodopsin molecule looking down on the cytoplasmic surface. And on the outside, you see little models of our nitroxide on the outside of each helix. And now this is a set of spin labels that we use pairwise to measure distances. So we select pairs from this set and do DEER to measure the distance over this long range with high resolution. Now, if you do the analysis right, you have a lot of data. It turns out from here you can determine the probability distribution of the individual nitroxide electron spins. And that's what these blue contours you see here. And overlaid on those blue contours determined experimentally is the crystal structure and predictions from the nitroxide models. So they're in extremely good agreement, except for this one. The crystal structure says they're here, the spin label says they're here. And it turns out this is because it's a really flexible sequence that's distorted by crystallography. So the crystallography is not right on that point. So this is the inactive state. When you flash light on it, what you see is that when you repeat this experiment, after you activate your Rhodopsin, this is what you see. So the sort of yellow-orange contours are now in the active state. And the only thing that happens is there's an clean outward movement of 252 in this helix TM6. And a smaller, this is 5 angstroms now, and a little over 5. And there's an inward movement of this helix 7 that by and large, nobody else is going anywhere. So it's a remarkable thing. This whole arc of the molecule is stationary. There's just this one helix and a neighbor that have a bit of motion. That opens up a cleft in the top of the molecule where transducin can bind. Now, Gobind liked to call this the central dogma of receptor activation, and he was right. It turns out the Rhodopsin is a member of more than 1,000 other receptors, and the experiments that have been done in many of them show that there's a similar or related kind of motion in all of them, a rigid body sort of tilting to the side. Now, so what I've just shown you is from the DEER experiment. Now, the DEER reveals static structures. This is the inactive state, and this is the active state. And it's static because it's a cryogenic experiment. These things are trapped and frozen at 100 degrees Kelvin. It's like crystallography. But what would happen if you would look at room temperature, at ambient temperature, in native phospholipid bilayer? What would you see? Well, we can now do that. I won't have time to tell you any of the details of the experiment, but I would like to share the result with you. And that is, if we look at spin labels around helix 6 and in the neighborhood of helix 6 and 7 and so forth, using these new techniques I just briefly alluded to, pulse saturation recovery and high pressure EPR, we can see that under ambient physiological conditions, that helix doesn't just switch out by 5 angstroms. The helix is actually fluctuating between a number of states. And here it says that the lifetime of each one of those states is 100 microseconds. We now know greater than 100. We now know it as many milliseconds. So this protein is fluctuating, presenting to the world different faces, each one which has a lifetime on the order of milliseconds. And you say, why is that significant? Well, we don't know yet, but it might be. It might be that Rhodopsin is presenting a different interacting surface, interact with different partners, promiscuity receptor. One last thing I want to point out that's related to this is sort of what I call conformational flexibility, or what anybody would call conformational flexibility, on a millisecond time scale. But there's another flexibility that is very interesting. That's on a nanosecond time scale. And that's shown here. So here's a ball of activated rhodopsin. I've tilted TM6 out by 5 angstroms. It's in the bilayer now. And this little red sequence you see here is a sequence that connects TM6 to its neighbor, cytoplasmic loop 3. And it turns out, measured by spin labels, to be highly flexible on a nanosecond time scale. So this thing is fluctuating around, flopping all around in space within the lifetime of one of those other conformational states. Now, the sort of punchline about this that makes it interesting is this turns out to be a sequence that's involved in the recognition of transduction. So, and together with Heidi Ham, we've actually explored the G-alpha subunit completely, actually, in many places, of transducin. This is the crystal structure of transducin. Here's the beta subunit, the gamma, and the alpha. Now, in exploring this, we found one sequence that's totally disordered. It's intrinsically disordered, and that is the C-terminus of transducin here. Now, the significance of that is that's the sequence that recognizes rhodopsin in the active form. So that these two highly flexible sequences sort of come together and find each other to form a more ordered complex, not completely ordered, but more ordered. There's a huge loss in entropy in this process. So you can ask yourself, why in the world would you ever design your recognition sequences to be highly flexible and disordered? Okay, I'll just tell you one of several really interesting reasons, and it's that you want high selectivity. You want these things to be really selective. You don't want any false recognition. And that usually requires you make many, many, many bonds, right? Many contacts. But when you make many contacts, you have a high affinity, which you don't want, because this thing has to now come off. So how do you do it? You make it flexible, because now when you make many contacts and you order it, you lose so much entropy that it reduces the free energy binding, so you now have a low affinity but high selectivity. That's one of the, probably the major regions in this thing. And while this is, the Grim Reaper has not risen yet, where is he here? So I guess I haven't. So I'll point out one more thing is that these purple sequences are also very interesting. These are allosteric coupling sites that couple these flexible recognition sequences to the active site of the respective proteins. In the case of rhodopsin, it's 11-cis red now, which is down below, it's not shown here. So when that's activated, this helix moves out, it moves the highly flexible sequence. And in this case, this flexible recognition element is connected by another alpha helix to the active site of the G protein, that is the GDP site that has to be rooted out. So when this interacts with rhodopsin, it moves that allosteric coupling agent and opens up the site. Okay, well, so it's a common theme, that's the extreme flexibility of these proteins, which is kind of a surprise to me initially, both on the conformational level and on this backbone level. So I'll finish just by telling you who did all this stuff. So everything that I talked about was done, except for the transducer work, in collaboration with Gobind over this period of time that was really fantastic for me. And many of the people that did the work were here. Kevin Ridge is here, John Wong, Dave Ferrens, Judith Klein, now Judith Klein sees Rollin, and Jerome Kim I saw in the audience. So Christian Altenbach was really the key person in my lab that sort of carried this out, doing EPR kind of work on this. I just so briefly mentioned these are the people that were involved in the technology development that I briefly mentioned. Okay, I will stop. Thank you.